Hi, everybody. This is going to be a lecture over chapter 3. This is introducing the tools of microbiology. These are your learning outcomes, your learning objectives for this chapter, and what the goals are for what you're supposed to be learning for this chapter, for those of you who are interested in this sort of thing. Okay, so first I want to introduce to you guys the concept of the 5 I's. So the 5 I's refer to inoculation. Okay, again, every time. If I could just remember it one time. Okay, inoculation. This is just referring to taking a sterile medium of some kind, and that's just talking about the stuff that we're going to be growing the bacteria in, for example. So taking something that's sterile, has nothing living in it whatsoever, and introducing something that we're interested in. So a swab of your patient's sore throat, or their swab of like maybe in their nasal cavity, whatever it is, a sample, stool sample, something like that. You need to grow it out. We're going to inoculate media first. So that's inoculation, introducing, sample into a sterile media in order to grow it up. Next, we have incubation. And these have to be done in order, FYI. So, and you do need to know what these 5 I's are. So incubation, that's just, it's not, if we put it into a media, it's not necessarily going to just grow just sitting on a countertop. Some of these organisms, actually many of the pathogenic ones, need to be grown at a warmer temperature in order to facilitate their growth. So keep that in mind. So that's incubation. So putting it into a situation where it is more likely to grow. So the temperature that it likes, how much oxygen it may need or not need, that sort of a thing. Then we have isolation. So isolation is referring to, if I were to take a culture of your throat, you probably have more bacteria. So if I were to take, hey, let's say you're healthy, and I take a swab of your throat, healthy person throat, and I, streak it on a plate, a general growth plate. Do you think you would grow anything, even though you're healthy? Yeah, you would. There's tons of bacteria in your throat, just naturally, right? That's common. That's part of your normal microbial biota. So how do you separate the pathogen from the normal bacteria? That's isolation. So trying to get separated colonies, whereas each of these little dots on the plate that it's showing on this figure, all these little dots in here, each of those dots is a colony, and that colony grew up from one single cell. So now we know if we can take that one colony and grow it up into media that was sterile, had nothing in it, then we know we're growing up a clone of just that one thing that we're interested in. So then we can test if there's a lot of different kinds of colonies growing on there. We can test each one individually and see which one has the traits that we think our pathogen would have. Isolation, separating it from the rest of it, right? Then inspection, essentially looking at either the growth itself, the growth itself. You guys don't see the stuff that I do. I just realized the little buttons that I press and everything. I'm very clumsy, sorry. Inspection, looking at them in the microscope, stains, what media they can or can't grow on based on specific things that we've added to it to inhibit growth. Are they antibiotic resistant? Have they become so naturally, or do they have some sort of genetic, abnormality to them? That sort of stuff. So this is inspection, learning about all of the details of this organism that's going to help us identify it. And then we can identify it. So using all of the stuff that we were able to gather in all of our investigation and bringing it all together and using the information to determine exactly what this organism is and its requirements and everything like that, so that we can start to treat it. Okay, inoculation. So going to the first I. Inoculation, we're going to prepare a culture using sterile media and a nutrient medium. So just in general, you're going to start with something probably that can grow most things, because you don't know much about your unknown causative agent. Medium is the singular and media is the plural. Technically, I say media probably all the time, and I won't blame you guys if you do too. So this is a nutrient-containing environment where microbes can multiply. So it is optimized for hopefully growth of whatever microorganism you could be interested in that moment, based on what you know about the organism. So sterile is a term that's referring to, and I said, it's free of all growth. So it should have no living things, no spores, no viruses, nothing. It should be completely sterile. So that way, and so should anything that you're going to be using as a tool to investigate it. So like in the lab, what we use are like our loops, our inoculation loops, and our inoculation needles and things like that. So those things should be sterile also. So we're not introducing any other outside, bacteria that could contaminate the investigation. So incubation refers to growing up these organisms, hopefully in the most ideal situation that we can for whatever we're studying them. Most organisms are going to grow between 20, which is about room temperature, 20 degrees Celsius, up to 45 degrees Celsius. Incubators can also, and the body is about 37 degrees Celsius, by the way. Incubators, we can also control gas. So that would mean oxygen and carbon dioxide levels, depending on the organism. They might be picky about something like that. Then we have microbial growth in liquid medium. We can determine if something is growing based on the cloudiness, whether there's sediment of some kind, some scum maybe at the top, or changes in color, something like that. All of those would indicate growth in the medium. And then we have on solid medium, those visible little dots that we showed on the previous slide. Those are our colonies. They're masses of piled cells that originate from one single cell. Many organisms grow in what we consider a workable amount of time, 1 to 2 days, including the ones that we work with in the lab. If we look at Mycobacterium tuberculosis, so tuberculosis, in order to culture this up to a workable growth, we would need 28 days. So and almost an entire month. It's a whole February at least. So in order to grow that up. And then you can see the differences in temperature preferences. So these guys will grow at lower temperatures than strep pyogenes, so that's strep throat. And then Mycobacterium tuberculosis. Jeez. We need to provide all of the nutrients that those organisms need in order to grow in whatever media you're growing it on. Sorry. Some organisms only need a few simple inorganic compounds. They're not picky. Other ones might need specific things that maybe they can't even make for themselves. Those sorts of things. Those would be growth factors. Right. Some organisms can't be grown in like media. They have to be grown in organisms, like actual living organisms. So that's something to be aware of as well. There's different kinds of media that you can work with. We can classify them based on their physical state, their chemical composition, and their functional type. Be aware of these differences. The chemical composition is just referring to whether it's chemically defined or whether it's complex. So whether we know exactly what went into it and we added, these amounts of things exactly, and we know what's in it, or if it's, basic, like a brain heart infusion. So it's just some brain heart breakdown of powder. You don't know exactly what's in it, but an idea, but that's considered complex because we don't know. It's general. So then we have the physical state. This one's a pretty obvious one, whether it's liquid or solid or something in between, or whether it can switch between one or the other, depending. And then the functional type, what you would be using it for. There is media that we consider general purpose to grow a whole lot of things. Enriched media, these contain growth factors for fastidious organisms. And fastidious means that they need a growth factor. It just means that they have some sort of nutrient that must be provided for them that they cannot make. We are fastidious, by the way. We require growth factors if you were going to grow us in a culture. You can't make a lot of your vitamins. You need to provide them provided to you in your diet. That includes like vitamin C, and that's why people get scurvy, because if they don't get vitamin C in their diet. Just an example. So exactly like that. That's what fastidious is, and that's what a growth factor is, vitamin C. Selective, differential, reducing, specimen transport, assay, and enumeration. Most of these kind of speak for themselves. We're going to go through most of these. A lot of these can serve more than one purpose, whether they can be both selective and differential, both enriched and, differential, that sort of stuff. I don't know. I feel like you guys kind of get an idea of liquid media, right? So this is going to exist as a liquid, typically broths or milks. Semi-solid, these have a clot-like consistency at room temperature, but they do not have a firm surface. We usually use these for motility assays to see that if the bacteria are going to grow. So it has a sort of firmness to them, but it's liquidy enough that the bacteria can move freely, and we can see if they did so. Then we have solid media, pretty straightforward stuff. Solid surface, they can get discrete colonies growing based off of one cell dividing. We do this for bacteria and for fungi. And for that reason, for those media, the ones that have to be solid surface, anytime that we have to solidify a lot of things, we're going to add agar to it. And this is a polysaccharide, again, made up of monosaccharide, subunits. That these are solidified solid at room temperature, and we can autoclave them. So that's how we sterilize a lot of stuff in microbiology as well as in hospitals. So we sterilize our media by autoclaving it, and it will definitely get liquid in the autoclave. And then we can keep it warm at about 42 degrees Celsius, and then we can pour it onto the plates and then let it cool to room temperature, and it stays solid. It's pretty neat stuff. Anything with 1 to 5% agar added into it is going to be considered, agar. You would say this is, blah, 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 agar. Okay, so defined media or synthetic media, we know exactly what's in it. Yeah, use it for research because we know exactly what's in it and we can say what's in it. For complex media, we have at least one component that's not chemically definable. It's just a general concept, essentially. So we have extracts of animals, plants, or yeast, like brain heart infusion. Like, what? So you know where it came from and you know what it is, but you don't know the exact chemical makeup of it in that moment. So that's what that's talking about. Okay, so we right. Enriched media and general purpose media. So general purpose media can grow just a variety of things. We try to make it as hospitable as we can for a wide variety of organisms, make it friendly, make it even baby friendly for fastidious organisms. Then we have enriched media. These are, like we said, for fastidious organisms that need growth factors in order to live. They need specific amino acids or vitamins, like we do. Then we have selective and differential media. These are often used for isolation and identification. You can use it for both of these. I don't know. But it wouldn't necessarily be your first step unless you knew exactly what route you were going down with it. You might want to start with other things to investigate. Just because if you're swabbing someone's throat and you're trying to determine what the cause is and you grow it on a selective media, you might not be able to grow the thing that you're trying to look at, because selective media only grows specific organisms and does not grow other organisms. So you could be discluding the thing that's causing the disease just because you think it's this organism. That's what I'm trying to say. So keep that in mind. Don't get too hasty with the selective media. So there can be stuff to inhibit growth of some organisms or just to favor growth of organisms that can use this resource versus this other resource. That not everything can do that. Then we have differential media. In differential media, we are not inhibiting anything from growing. It should be, a wide variety of organisms can grow on this. However, if they can utilize this or that thing in the media, then the media that they're growing on, then they will produce byproducts that can usually cause color change in the media, often based on pH, by the way. So we have pH indicators added into the media, and then based if there's acid production or if there, is some sort of other byproduct made, we'll have a color change in the media or in the growth itself. So that's what we would see with differential media. we'll investigate that a lot in the lab. So blood agar is a differential media. It also tends to be an enriched media. So it can investigate hemolysis, the presence of hemolysis, and if your organism is able to break down red blood cells. If you can lyse or break down red blood cells, then you will see an effect on the agar itself, because this is literally blood agar. The beta hemolysis, you have complete lysis. So this is going to have a clearing around the actual growth. And then gamma hemolysis, you would have no hemolysis. I don't know why it's called hemolysis when it's not, but there's growth, but no hemolysis. Then that's gamma hemolysis. And then alpha hemolysis is you have a greenish change to it. So gamma, here we have, this is no hemolysis. We have beta here in these, you can see clearing. So this is complete hemolysis. So strep pyogenes, the thing that causes strep throat, does this. And look, patient A, their sample was able to do that. So they probably have, based on this anyways, strep throat instead of whatever this other strep organism, strep sanguinus. This does cause greening, but we don't see clearing. So this is alpha hemolysis. So this is incomplete hemolysis. Oh. So media can be selective, like we said, has inhibitory factors in it to keep certain things from growing, but it can also be differential, which wouldn't inhibit necessarily, but does cause color change, so you can tell the difference. Some media is both. So a great example of that is mannitol salt agar. It's really good for staph organisms. So if you have growth, like you can see here, I guess, I don't know, there's maybe a limited amount of growth. I don't know if there really is any growth, but there definitely is here. Anyways, here you can see that this turned yellow, right? Obviously, this is a yellow area. The other areas are a little bit more pink. That's the original color of the media, by the way. You probably didn't know that. I'm sorry, should have mentioned that. But the original media is pinkish color. And that's because it has a pH indicator in it. And if the organism can break down the mannose sugar, it creates an acid byproduct, and that causes it to turn yellow from the pH change. And that is what happens in the case of S. aureus. It turns it yellow. And so that is a differential versus the pink. So this could be S. epidermidis. So it's normal skin bacteria. Yeah, so it'll grow on that, but it will not necessarily be able to metabolize the mannose sugar and turn it yellow. It shouldn't be able to do that. So that's interesting. So we've seen that. So what makes it selective though? There is a high concentration of salt, and that makes sense too. Both of these organisms grow on the skin, and the skin has high salt. So that makes sense. With McConkey agar, we have the addition of bile salts. So organisms that live in the gut, which are known as enteric organisms, they will grow on McConkey agar, whereas other organisms won't be able to, like gram-positive organisms like staph epidermidis. They wouldn't be able to grow on McConkey agar because they can't grow in the presence of bile salts. So we've already got that selective aspect. Then they have a differential aspect where the growth on the actual media, like the actual bacterial colonies, will turn pink if they are able to use lactose. If they can metabolize lactose, they will turn pink. So that is differential. That part is not selective. So they're both. Okay, then we have reducing media. This is for anaerobic bacteria and aerobic. So aerobic is just like, if you're going to aerobics, you're probably going to be, working out and breathing a whole lot, right? Think of that. So this is going to be anything that's aerobic. That's O2. They need O2. Anaerobic. We'll just do this. No oxygen. They do not use oxygen. They have no oxygen. In fact, they can often die in the presence of oxygen. So if you're trying to grow something like Clostridium species, a lot of those are anaerobic bacteria. You would want to be growing those in an anaerobic environment, so no oxygen there, and special reducing media for that. Now we have carbohydrate fermentation media. That's just media that we use to see if certain organisms can metabolize and ferment, truly ferment specific carbohydrates. Okay, transport media, exactly what it sounds like. It's made to help, with transport. Assay media, specific media that are made for specific assays, like diagnostic assays or something. So they're designed for those assays. Then enumeration media, so trying to determine colony counts in certain things. Like when I did food testing, we did a lot of growing bacteria on specific media. Specific kinds of special, we used this stuff called Petri film, which was like if you had a plate but turned it into like a card. I don't know how to describe it, but yeah, you would grow stuff on that and you can count the actual number of colonies on them. So again, colonies, discrete amount of cells, came from one original cell. So that's important for isolating. So when we're trying to isolate, what kind of ways can we separate out the cells? If you have super concentrated sample, something like a stool sample, and you're trying to colonize, if you're trying to isolate, boy, that's going to grow up a lot of stuff if you just straight up grow it, right? So how are we going to get it diluted out so we can get individual colonies? The first method, the one we're doing in lab, is the streak plate. You start by placing your stuff down on your plate. Then you flame your loop to sterilize it, and then you spread it out, and then you can go back into that and then spread it out. These guys did it a little different than I did. I would have done it like this, and then you can go back at flame your loop and then go back in and squiggle it out. And then you should at the end have diluted out that sample enough by going back in and in and in and in, that now you can have individual colonies going on. So that's the goal there, individual colonies. The pour plate method, we're not going to be doing this, but it is a method that you can use. You're going to have tubes that you've grown up that you're going to be adding into Petri dishes with agar, essentially, and then you're going to dilute them out that way. So I'm going to describe this better picture. Okay, here, yes. Essentially you inoculate this tube and then you mix it in with some agar, pour out, and you can see colonies grow out. And then each of these are diluted out samples that we've put into there, so that we have diluted out and diluted out and diluted out until eventually you can get countable colonies, countable or individual for isolation or further studying. Okay. The spread plate, you use this little hockey stick guy. You take whatever, okay, there is a picture on this next one. You take your sample that maybe you've diluted out somehow in more broth or whatever, and you squirt a little bit on some agar, and then you spread it out with this sterile hockey stick guy. A lot of times these are disposable, and you spread it out and then incubate it and let it grow, and hopefully you get colonies. All right, so you can go back to those, and once you get your individual colonies, pick out one of them and grow them into a sterile media. That media that is now growing from that one colony, that is a pure culture. It is growing from a single known species or type of organism. We know that it's from one organism because it's from one colony. Even if you're not sure what it is yet, you know it's a pure culture. Anexic is a term that means to free of other living things except for that one thing being studied. And then subculture is a term that refers to a second level culture from a well-isolated colony. So maybe you spread it out again, you well isolate it, you culture it out again. That would be the subculture from that original sample that we got. Okay, that made sense. Mixed culture, we have two or more differentiated species of organisms. Man, people email me. And a contaminated culture. So a mixed culture is like if I go and swab your throat and we put it on a plate and a bunch of stuff grows. That would be a mixed culture. A contaminated culture would be like if we go and swab your throat and we spread it out on a plate, a bunch of stuff grows, but somewhere, whenever somebody went to go look at that plate while it was growing, they sneezed on it, a whole other person, then it could become contaminated with that other person's bacteria that was not supposed to be there in that experiment or in that patient sample or whatever it is. That's the difference there. So for inspection and identification, we have a lot of different things available to us as microbiologists to investigate the identities of these microorganisms, whether it is phenotypic, that means looking at certain observable traits of these organisms. That includes things like, media color changes or something like that. Genotypic, so doing something like PCR to look at the genes of these organisms. Immunologic, their reactions with bacteria, their reactions with antibodies. Macroscopic analysis versus microscopic. So macroscopic could be like looking at the colors of the actual colonies themselves, how the texture, what the growth and broth looks like. And then microscopic analysis, literally looking at the cell shape and other things like that. So biochemical tests can tell us about nutrient requirements of the organism, enzymes that the organism produces, how the organism makes energy, that sort of stuff. So we did mention this a little bit. This is just looking at the size of different organisms, whether we're talking about a louse like that, if you were having lice, and reproductive structures of bread mold versus these other organisms like Giardia lamblia, which is the protozoa that causes, diarrhea in often hikers who drink water that hasn't been treated. Versus a red blood cell, and then we have Saccharomyces cerevisiae yeast. It's like in bread. Anyways, we'll go down the whole way. We can see all the different size, like E. coli bacteria, mycoplasma, and then we can break it down into the viruses. Like poliovirus is a very small virus, the flagella of a bacterium, and how it compares to like poliovirus. Prions, which are literally just protein. We'll talk about those because those are interesting. And then we have the diameter of DNA compared to that and whatever, and then amino acids and then hydrogen atoms as far as that goes. So this is just showing you the scale of everything. This is not to scale. I want to be clear about that, but it is describing the scale to you. So sometimes if we want to look at these organisms, we need to be able to magnify them. That's the whole point of what we're talking about with microbiology. We're talking about microbes, things we cannot see with the naked eye. We need a microscope, so we have to magnify them. Refraction is a term referring to bending or change in angle of light ray through medium. And if we're going from one substance to another substance, that can cause refraction. So a greater refraction. So going between the air and glass, those have a very different, refraction between one another. So that can cause distortion of the image. Essentially, whenever we are looking through a microscope, what we're seeing is the virtual image. So the real image is what's seen by the lens that's picking up the image from the specimen itself. That is called the objective lens. And then it goes through, some mirrors and stuff, and then through another lens, through the ocular lens, that creates the virtual image that magnifies it just a little bit more. And then that's what we see. So that's what's really going on in the microscope. Typically, if you want to get the full magnification of whatever you're looking at, you will do your objective lens number and multiply it by your ocular. And yes, our microscopes do have these, and they do say their magnification amounts on them. So when you are asked questions about this on the test, this is not some sort of trick. So your objective lens could be like your 40x objective lens, and then your ocular lens might be a 10x. So in this case, your total magnification, this is multiplied. So it's going to be 400 400x. That's the total there. So whatever you're seeing in the image in your eyeballs, that's 400 times larger than what it really is. So these are the structures of a microscope, and the ones that are circled, you definitely need to be aware of how these work and what they are and all that. I would know all of the parts, FYI, because I would expect you guys to know that in part of the laboratory, but definitely the objective lens, what's going to be magnifying and creating that real image. Here, your ocular eyepiece, this is going to be the virtual image. And then whether your light control, your coarse focus, we only use this on the lowest objective lens, 4x. And then we have the fine focus, and then the stage adjustment knobs down here to move your slide around. Do not be moving your slide around with your hands. I better not see it. No. All right, so the main properties of an effective microscope: magnification, how big you're going to make the thing look; resolution, that's like the clarity of the image; and then contrast. You have as much resolution as you want, but if you have no contrast, it's going to still be hard to see your organism. If it's clear, unclear, just forget it. So we have the ocular lens, right? We just mentioned this. The virtual image is the ocular, the real image is the objective. The objective is what's going to be looking like right at the specimen, okay? So this is how the light is going to be traveling through the microscope, out of your source, get mirrored up through the light source here, and then filtered through the condenser itself onto the specimen, which is going to be magnifying that image through the objective lens for the real image. And then through some mirrors, mirrored back out through here. And then this image here will be the virtual image once it gets out of that ocular magnification. So we already went over this. This is that magnifying power. This is going to be multiplying the objective lens power by the ocular power. Resolution is the capacity of the optical system to distinguish between two objects. So the resolving power literally is going to be referring to our ability to distinguish things, even though they might be close to one another. So yeah, I don't want to say resolving power. It gives you more clarity. So this is just showing us a simple difference between, I don't know, these organisms based on what you can see from resolution. You might be able to see more characteristics as if you have low resolution versus high resolution. And we already know that, right? Because of TVs. So you can see a lot more detail. It's really all that's talking about. With oil immersion. So on our 100x lenses in our light microscopes in the laboratory, we have oil immersion lenses. These we will be adding oil to it so that the light, as it travels into the glass and then hits onto the specimen, as it's going into the lens and back into the glass of the lens, if we had air there, it distorts the image, but we put oil there because it has similar optical qualities to the glass. So that's the whole purpose of the oil there, so we can get better resolution. It minimizes light scattering. So we can see here with the oil, we have far less light scattering and light loss, essentially, of image quality when we have presence of the oil. Contrast is just talking about essentially refractive index and our ability to see between the, or it's harder to describe. You guys know what contrast is, I would hope, but our ability to discern whatever object we're looking at in the microscope from its background. That's what I'm trying to say. So we have 4 kinds of light microscopes: the bright field, which is what we use in lab; the dark field, which is like kind of what it sounds like. It's going to have a dark background for it. Phase contrast. We're not going to talk too much about that. Or interference. That also not going to talk about much about that. Then we have fluorescence microscopes that usually uses fluorescence fluorescently labeled molecules so we can shine certain lights on it and make them glow so we can see certain parts of the cell. And then confocal microscopes that use laser beams that often are paired with some fluorescence. Anyways, that would allow us to look into parts of the cell and create 3D images. I know nothing about super resolution light microscope that uses 2 lasers. I know nothing about that, but apparently that's a thing. Right, so when you're preparing your specimen to look at it under the microscope, you need to consider, are you looking at the organism's movement? Do you need it to be alive? Do you need to look at what certain things are you trying to determine about your organism? Are you trying to compare it to other organisms? What are you trying to know about these other organisms? And so on and so forth. So, thinking about all that, whether you want to do a wet mount or if you want to do, a heat fixed smear, for example. So wet mounts, we're going to, what they sound like. You take a drop of your broth, you put it onto a slide, you put a cover slip over it, and you look at it. You don't get to have typically any stains, so there's usually not a whole lot of contrast there. This can damage larger cells like eukaryotic cells. They are susceptible to drying out. It actually happens a lot more quickly than you would think. But they are very quick to repair, and they cause a lot of contamination. They really do. They kind of get all over the place, and it's gross. So when we do that, be sure to clean your microscope. Be very much a stickler about that. A hanging drop is kind of similar to the wet mount, except we have a special slide that has a little divot in it. I'm gonna put a little bit of Vaseline around it so that it doesn't dry out as fast. Okay, so these are our stained smears. So our smear technique, you're just taking like your broth or a little bit of colony or whatever and spreading it on a slide and letting it dry. That's a smear. You can go farther and do more than that. You can fix your smear. So we can heat fix it, or we can fix it with alcohol or formalin. Most of the time we would be heat fixing it. It's literally running your slide over a flame a few times. This will kill the cells, secure them to the slide, and preserve the cellular components of the cells so that they are accurate when you stain. We get a lot of contrast from staining. That's like the #1 reason we stain cells, so that we can actually see the features of the cells. Dyes will impart colors to the cells, obviously. And then we have 2 kinds of stains that we can use in staining procedure. I don't know how to word that, but that's what you get. Basic and acidic. So the basic ones, or cation ones, these have positive charge. They're going to be attracted to the negative cell walls. Now remember, those cell walls are going to be having phospholipids, and the phospho group is what's on the outside, so it's negatively charged. So the basic stains will want to stick to the cells themselves. The acidic stains, the anionic stains, however, they have a negative charge. They're going to be repelled by the negative on the cells, and so they're going to stain the background typically. So that leads us to calling them positive stains that are attracted to the cell walls to get the cell color, and negative stains that are going to be in the background, repelled by the cells themselves. So just looking at some negative stain and positive stain, as far as like what they would look like whenever you're doing the stains, and what subtypes of stains would be performed for something like this. So we will be doing positive staining and negative staining in the lab, both of them. When we do our capsule stain is when we're going to be doing our negative staining, and we will be using Congo red for that. But anyways, there's all these different kinds of stains. We're going to be doing quite a few of them in the lab. So a simple stain is just going to have a single dye. It isn't what it is. We're just staining the cells to give contrast to look at them. We don't need to know much else. Otherwise, we will be doing differential stains. Usually involves several steps in the staining procedure. Maybe an initial primary stain that interacts with whatever thing you're interested in about that cell, and then a rinse, and then a counter stain. And so whether or not you're positive or negative, you might need to be, counter stained to see if you're negative or not. So this is going to include things like the gram stain, the acid fast stain, and all this. So whether or not they have these things can tell us whether or not we have these things based on how we do our staining procedure. So the Gram stain, this was developed a long time ago to discern between gram-positive and gram-negative bacteria. Gram-positive bacteria stain purple, gram-negative turn red or pink. So positive is purple, positive is purple, positive is purple. And then I always think negative is red, negative is red, negative is red. It doesn't matter, whatever works for you, I guess, but that's what works for me. And you're going to need to know this for sure. This is a biggie. So gram stains. These are determined, you're positive or you're negative based on your cell wall composition and your cell envelope of your bacterial cells. Next is the acid-fast stain. This just determines if you have the presence of the waxy mycolic acid on the outside of your cell. This is going to be testing for mycobacterium. So things like Mycobacterium tuberculosis or Mycobacterium leprae for leprosy. So these guys are going to stain pink from the stain, the carbofusion stain. And then we counter stain with methylene blue. That's the negative cells. So the negative cells are blue on this one. For the endospore stain, vegetative cells versus endospores. The endospores, the ones that we're going to be doing in lab, are going to be green endospores, greenish bluish endospores. And then the vegetative cells themselves are going to be red with saffron. So that's going to determine if your bacteria have endospores, and the bacteria that have endospores are going to be from the genera Bacillus and Clostridium. So that's what's going to be happening on those.
Thank you very much.
Yep, thank you.
Thank you. Good day.
You too.
Okay.
Sorry about that. Okay. Put notes up to let him know, but he still comes in. So he's nice enough. So he means well. So anyways, right. Endospore stains. So endospores are like protective structures for bacteria. And we will, don't worry, talk about those plenty when we get into the bacteria chapter in chapter 4. So don't worry. Don't worry. Okay, then we have some special stains like certain spore stains and capsules that are differential, but also fall into a special category because they pinpoint specific characteristics, like the presence of an endospore. I don't know. That's going to look at, like when you look at an acid-fast stain, that's not necessarily going to be a special category because we're not looking to see if an organism has a special feature going on with that cell. That's what's special about the endospore. Does it have this special thing? Does it have the endospore? Does it have a capsule? Okay. Okay, the capsule stain is just looking at, basically we have a stain of the bacterium, and we have a stain of the background, our negative stain, with positive and negative typically. And then in the middle, you see how there's white around the cells? That is the actual capsule itself, and it repels all stain. So that's why we see that white around halo around it. If it were just cell and no capsule and just background, it would be cell color and the background right up against each other. So that's the difference there. Flagellar staining. This is, flagella are what bacteria use to move around. So we can perform specific stains that help us visualize the flagella a little bit better, so we can understand better their orientation of the flagella around the actual bacterial cells. Yeah, and that can be helpful in determining the identity of an organism. Some bacteria only have one flagellum. It's like this. A flagellum is a single one. So if you were like this, that's just one. Clearly, this is it all over it, right? This is what we call peritrichus. We'll talk about that more soon. So that's that for this chapter. Our chapter question is going to be, when you are trying to isolate several separate bacterial species from a mixed culture for identification, you don't know what you're looking at, and you're not sure what your causative pathogen is or something like that. Why might you want to avoid using selective media at least at first during the isolation phase. We did talk about that earlier, so good luck with that, and I will see you guys for the next chapter.